Examining peanut response to photointensity in speed breeding. Ty Seeley, Jeff Dunn, David Jordan, Andrew Oakley, Crop and Soil Science Department, North Carolina State University, Raleigh, North Carolina. Ricardo Fernandez, Christian Collado, Horticulture Department, North Carolina State University, Raleigh, North Carolina. Abstract. Peanut, traditionally a long season crop, creates challenges in breeding because of slow generation time. Speed breeding, a multifaceted approach using marker-assisted selection and controlled environment grow-out techniques, can help decrease generation time and reduce time for cultivar development and release. Arachis hypogea, Bailey 2, was exposed to differing light intensities in order to determine if photointensity has an effect on peanut pod set, seed count, and post-harvest germination percentage. Data will be utilized for application in a peanut speed breeding program, which aims to decrease time between generations, allowing for faster cultivar development and release. Objectives. Decrease cycle time to increase genetic gain by examining different light intensities and their effect on plant and pod development. The left picture shows a plot after 59 days of growth. The right picture shows a plot after 38 days of growth. Materials and methods. Bailey 2 plants were evaluated at 0, 200, 400, and 600 micromoles per meter squared per second of additional light provided by 2 LG LED panels over each treatment. 4 harvest intervals, 70, 80, 90, and 100 days, were employed in each treatment to see whether photointensity influences time to flowering, pod production, pod maturity, etc. The trial was set up in a randomized complete block design with 3 replications. 2 plant subplots were nested within each of the 4 zones, light intensities, organized within each block. Seeds were planted on April 29, 2022. Initially, day and night temperatures were consistent at 30 degrees centigrade to prevent excess stretching, but night temperatures were later reduced to around 25 degrees centigrade. A volumetric fertigation system was implemented after plant establishment with a custom soluble fertilizer blend. Fertigation was activated after reaching a soil moisture content below 30% by volume and ran for 10 minutes to reach field capacity of 40%. Calcium concentration and fertilizer blend was increased from 69 parts per million to 200 parts per million 1 week after all plants had flowered in order to ensure enough surface level calcium was available for pod fill. Results. While the experiment is ongoing, preliminary results show flowering times occur sooner and higher light intensities, shown by the flowering heat map. Colors signify number of days after the first plant flowered, day 0, given in dark red. Additionally, plant height over the first 3 weeks after transplant shows that as light intensity gets lower, plant height increases. On the far left, we see a key with colors ranging from dark red to black in the first part and the upper part, each correlating with a certain number of days after the first day of flowering. The bottom part of the key shows 4 colors which correlate to different light intensities. To the right of that, in the middle, we have a plot map showing the flowering heat map of all the zones and plants in the greenhouse. Each bordering color around each of the zones represents each of the light intensities. Light red represents 0 micromoles of supplemental light. Light green represents 200 micromoles of supplemental light. Orange represents 400 micromoles of additional light. And purple represents 600 micromoles of additional light. In the upper right-hand corner of figures, we have a map of the greenhouse showing all of the plants. And below that, in the bottom right-hand corner, we have a graph showing average plant height by intensity during first 3 weeks after transplant. And this shows that the tallest plants are 0 micromoles. The second tallest plants are at 200 micromoles of supplemental light. The third tallest are at 400 micromoles of supplemental light, and the shortest plants are given 600 micromoles of supplemental light at 3 weeks after transplant. Discussion. Less pegging in this experiment is happening than anticipated. Literature shows photoperiod has effect on pod production, but further evaluation may be needed. 20-hour photoperiod has proven successful in other preliminary experiments, so shortening of photoperiod in future experiments may provide better pegging results. Increased plant height seems to be correlated with lower light intensity, but data still needs to be analyzed. Plant height could increase with lower light intensity due to shade avoidance responses. Results from this experiment will be used for more applied grow-out applications in our own greenhouse space to refine techniques. In addition to the photointensity study, in future experiments, marker-assisted selection will be utilized in parallel with this speed breeding approach. Marker-assisted selection will allow for materials to reach breeding benchmarks quicker while increasing the frequency of beneficial alleles within the breeding population, thus improving genetic gain. The accessions IAC 322 and SPT 1012 contain a late spot resistance Arachis cardenacei introgression block on chromosome 13. Successful crosses can be validated within 2 weeks of seed germination to see if the desired block was introgressed. Selfed plants and hybrids that do not contain desired blocks can be discarded.